Okay, so we're diving in today,
Yeah.
looking at this file, what our sources are calling a coming catastrophe. It seems less like an if and more like a when.
Right. We're talking disease X, but maybe from a different angle this time.
Exactly. It's through this lens
Uh-huh.
of exponential risk, this massive pre-existing global crisis. Antimicrobial resistance, AMR.
Ah, okay. So not just the new virus, but the environment it lands in.
Precisely. And our mission today, it's driven by this AI-generated sort of mandate a listener shared. We really need to get our heads around why the systems we have now.
The surveillance, the testing,
Yeah.
all that.
Yeah, all of it. Why it's almost, guaranteed to fail when the next big pathogen shows up.
And there's a solution proposed.
There is. We have to unpack this idea of deploying a specific AI system, Dr. Maya, before things get bad, preemptively.
Okay. That sounds urgent.
Yeah.
What's the headline warning from the source material then? What's the big number?
Well, this is what jumps out. The claim is the next pandemic collision, they call it. It's likely to be 20 to 30 times worse.
20 to 30 times. That's huge. Why? What's the multiplier?
It's entirely down to AMR. The fact that we're basically stepping into what they call a post-antibiotic world.
So our defenses are already down?
Yeah.
Before the main attack even starts.
Yeah.
Is that the idea?
That's the core insight, yeah. It's not just about some new virus-jumping species. It's about that virus hitting us when our safety net, treating secondary bacterial infections, that net's already ripped to shreds.
Mm-hmm. Can we put some scale on that AMR crisis?
Yeah.
Just so people grasp it?
Okay, yeah. Globally right now, AMR is directly killing 1.27 million people every single year.
1.27 million. Okay, that's already significant.
It is, but the projection,
Oh,
that's
yeah.
the terrifying part. If we stay on this track, by 2050, AMR is projected to kill 10 million people a year.
10 million annually.
Annually. And that number, 10 million, would definitely push it past cancer as the world's leading cause of death.
Wow. Okay, so when you put those numbers out there, the idea of needing a proactive solution like this Dr. Maya you mentioned, it starts to make a lot more sense.
Right. Because our traditional approach, it's reactive. Find the sick person, test them slowly, trace their contacts. It's always behind the curve.
Especially with something multi-resistant that moves fast.
Exactly. So the argument is we need to build this new infrastructure now, not, wait for the panic when it hits.
Okay, let's establish that foundation then, this post-antibiotic reality.
Yeah.
How? How did we even get here? How are bacteria starting to win?
Well, the sources point to, it's a natural process, resistance. But human activity is, put it on, fast forward.
Right, it's biology, but we've accelerated it. How?
Misuse and overuse of antibiotics everywhere. Human medicine, absolutely, but also agriculture, livestock. It's sort of saturated our environment.
So we've essentially been training the bugs, selecting for the toughest ones.
That's it. We've selected for the strongest, most resilient bacteria, and that's led us straight into this era where drugs we've relied on for decades, they don't work anymore.
The source used an analogy, didn't it, to make it more personal?
Yeah, the sepsis casket. It's pretty chilling. Think about everyday injuries. Your kid scrapes their knee at the park. Or, your grandfather gets a little cut while gardening. Maybe you pop a pimple.
Things we don't even think twice about.
Exactly. In the antibiotic era, no big deal. A quick course of penicillin, maybe, done. But now, post-antibiotic era.
That's where the trivial becomes potentially lethal.
Right. If the bacteria getting into that little cut are resistant to the first antibiotic and then the second, the infection just runs wild.
And the body's response, that's sepsis.
Yeah, the immune system goes into overdrive trying to fight this widespread invasion. It triggers massive inflammation, organ failure, blood pressure crashes, sepsis.
Which links back directly to past pandemics, right?
Yeah.
Like 1918.
Absolutely. Think about the Spanish flu. Most people who died then, it wasn't the virus itself that killed them directly.
It was the secondary infections.
Pneumonia.
Bacterial pneumonia, exactly. And doctors back then didn't have effective treatments for it. Now we have antibiotics. But if they fail because of AMR, we're structurally, back in 1918, facing a virus, but unable to handle the bacterial follow-up.
That's the terrifying parallel. We're facing the same outcome, but this time it's because our miracle drugs have stopped working.
And this isn't theoretical. We're seeing highly resistant pathogens emerge now, right? Like that super fungus.
Candida auris, yeah. And it's not even a bacterium, it's a fungus. An emerging one causes invasive disease, especially in people who are already very sick. And the mortality rate,
Over 40%, the source said.
greater than 40%. It's staggering. And the real problem with C. auris is Well, it loves hospitals.
It thrives in healthcare settings,
Right.
long-term care facilities, too.
Exactly. It sets up shop, causes these prolonged outbreaks that are incredibly hard to control. It basically becomes endemic, a permanent resident in the places meant to heal people.
And the resistance profile is bad.
It's why they call it a superbug. Resistance to fluconazole, a common antifungal, is up between 87% and 100% globally.
Almost total resistance to a frontline drug.
Yeah, and even worse, they've found pan-resistant strains.
Pan-resistant, meaning resistant to everything.
Resistant to all three main classes of antifungal drugs we have. They've seen these in the US, South America, India. It's global. This thing has defenses against our entire current arsenal.
Wow, okay. And it's not just the hospital environment creating these things, is it? The One Health idea suggests broader environmental factors are at play.
Right. Zooming out, you see how our activities, our environment, are sort of actively designing these superbugs, like the hygiene paradox.
Oh, explain that one. We're too clean.
Well, the hypothesis suggests that by creating these super sterile environments, especially for kids, we might be preventing their immune systems from learning properly. Not enough exposure to everyday, mostly harmless microbes.
So the immune system gets twitchy, overreacts to non-threats.
Yeah, trigger happy is the term used, leading to things like allergies. It's this weird contrast when you look at ancient populations.
Like the ones in Nubia and Egypt.
Exactly. Research on skeletons found traces of tetracycline. The idea is maybe their diet, perhaps from fermented grains or something in the soil, gave them this low-level, constant exposure to antibiotics.
And that might have actually been protective, helped keep infectious disease rates low.
Potentially, yeah. Whereas we actively remove those kinds of exposures and then, ironically, introduce things that accelerate resistance.
Like disinfectants.
Bingo. Our obsession with cleanliness, especially post-COVID, the massive increase in using biocides, specifically these things called quaternary ammonium compounds, QACs, homes, businesses, hospitals, they're everywhere.
And QACs promote antibiotic resistance. How does that work?
Okay, so it's a bit technical, but basically when bacteria encounter QACs, they have to defend themselves. They switch on these pumps.
Pumps, like to pump the disinfectant out.
Exactly.
Yeah.
Multi-drug efflux pumps, genes like MEXCD, OPRJ, and AQRB. Think of them like bilge pumps on a ship, constantly pumping out the toxic stuff, the disinfectant.
Okay, got it. So they pump out QACs.
But here's the kicker. The same pumps they use to eject disinfectants, they also use them to eject antibiotics.
Oh, so by making them resistant to the disinfectant.
You're unintentionally training them to resist antibiotics, too. Multiple classes like quinolones, macrolides, at the same time.
That's not good.
It's worse. The genes for QAC resistance and the genes for antibiotic resistance are often physically linked together on these things called mobile genetic elements.
Mobile elements.
Yeah.
Like little transferable packets of DNA.
Precisely. They can jump easily between different bacteria, even different species.
Sharing the resistance instructions.
Exactly. And where do all these bacteria, disinfectant residues, and mobile genetic elements end up mingling?
Uh-oh. Wastewater treatment plants.
You got it. The source is called WWTP's hotspots for developing and spreading resistance. We're basically running a massive distributed superbug factory and then flushing the results back into the environment.
Okay, so connecting it all back. The environment, our cleaning habits, the baseline AMR, it's all setting the stage, making those secondary bacterial threats potentially unmanageable in the next pandemic.
That's the terrifying synthesis, yes. Which makes our current pandemic strategies seem, well, incredibly inadequate, given that reality.
Yeah, that's the logical next step. We need to really pick apart why the current approaches are failing, starting with testing and tracing.
Right. Mass testing. Sounds great. Sounds robust. But as a containment strategy, the sources say it's basically a fantasy.
Why? Doesn't testing tell you who's sick?
It only tells you if someone is infected right now. To actually stop a fast-moving bug, especially one with asymptomatic spread, the source argues, quote, you would have to test everybody every day.
Which is, yeah, logistically impossible on a global scale.
An absolute illusion of security. You'd always have huge gaps, delays. Detection would lag way behind the spread.
And the tests themselves aren't instant or always perfect, are they? There are bottlenecks.
Big time. Standard culture tests. They take days. And they're not always sensitive enough. Remember, CRS. Nearly half of those invasive cases are culture negative.
So the standard test misses half the cases.
Roughly, yeah. If you want fast, accurate ID, especially for these priority bugs, you need DNA sequencing. And that's usually only available in specialized reference labs. More delays.
Delay seems to be the killer here. That lag time costs lives and it makes containment basically impossible. What about contact tracing?
Same problem, fundamentally. It's too slow. Even during COVID, with massive efforts, the median time from collecting a sample to notifying contacts was around 6 days in many places.
6 days. An infected person could have spread it significantly by then.
Easily. Multiple new clusters seeded before tracing even catches up. The sources flatly state there was room for improvement in timeliness.
And that's just the speed issue. There were other problems, right? Privacy concerns.
Huge ones, and often justified fears about civil liberties, data misuse. When people don't trust the system, they don't comply. That undermines everything for the next time.
So failures in detection and tracing. What about higher up? Governance, countermeasures.
Yeah, the failure isn't just on the ground. Our whole approach to developing medical countermeasures, MCM, so vaccines, drugs, diagnostics, it's overwhelmingly reactionary.
We free for the fire, then buy the insurance.
Perfect analogy. We wait for a pandemic to hit, then we ramp up R&D for that specific bug. The data's clear. Look at influenza research, the Orthomyxoviridae family, publications, clinical trials. Huge spike only after the 2009 H1N1 pandemic.
We're always chasing the last crisis, not preparing for the next one.
Always playing catch up.
Yeah.
And even when we do develop the MCMs we need, the supply chain is incredibly fragile.
Fragile.
Okay, get this. Analysis of MCMs for what they call viral pathogens of pandemic potential, VPPPs. 30%. 30% of these critical drugs or supplies have zero domestic manufacturing.
Wait, zero. So for nearly a third of essential pandemic tools, we're entirely reliant on other countries during a global crisis when everyone needs them.
Exactly. Think about the geopolitics of that. And it gets even scarier. Of all the available MCMs studied, 51%, over half, are produced at a single manufacturing site worldwide.
One factory. For half the world's supply of a critical countermeasure.
One single facility. Imagine if that factory's in an earthquake zone, or a floodplain, or there's a fire, or local unrest, or even just a major power outage.
Half the global supply.
Oh,
Gone.
vanishes. The lack of redundancy it's almost unbelievable. It guarantees massive disruption right when you need stability the most.
And that level of vulnerability just screams for decentralized solutions, doesn't it? Things that don't depend on one factory or one central lab.
Absolutely. And then there's the policy side, the social failure. Think about the non-pharmaceutical interventions, the NPIs during COVID, lockdowns, masks, distancing.
They drew a lot of criticism.
They did. Sources mention criticisms like arbitrary, capricious, particularly when implemented inconsistently, and some measures were just ineffective. Temperature screening is the prime example.
Right, the forehead scanners everywhere.
Yeah. Widely used at airports, universities, building entrances. But the studies found sensitivity was incredibly low, sometimes as low as 0% to maybe 40% in universities.
Why so low?
Well, two big reasons. Asymptomatic carriers don't have a fever, and simple things affect readings like being out in the cold or having just walked quickly. It's easily fooled.
So it gave a false sense of security while missing most actual cases.
Declared insufficient as a primary means of detecting COVID-19. And relying on these flawed metrics just eroded public trust even more.
Okay, so testing is slow, R&D is reactive, supply chains are brittle, policies can be flawed. Are we missing any vulnerabilities?
One more big one, hiding in plain sight, especially concerning AMR, the hospital environment itself. We tend to think of pests, insects, rodents as just annoyances.
But they're more than that in its context.
The sources identify them clearly. Reservoirs, vectors, and even sentinels for antimicrobial resistance genes. Flies, cockroaches, ants, rats, mice.
They carry resistant bacteria.
They do. They share our environments, obviously, including hospitals. Research specifically shows ants, flies, other insects carrying antibiotic-resistant bacteria inside hospitals. They can transfer these resistant bugs through direct contact or by contaminating surfaces, food.
So even a supposedly sterile environment is constantly being seeded with superbugs by ants.
It's an underestimated hazard, yeah. It implies our standard pest control, our hygiene protocols, they might be completely outdated for the AMR era. We need to rethink that. Maybe UV light systems, different biological controls.
Wow. Okay. So if our current systems are fundamentally broken, slow diagnostics, fragile supplies, policy missteps, hidden reservoirs, it really paints a picture of being unprepared for that decentralized post-antibiotic disaster you mentioned earlier.
That's the core argument, which leads us to the proposed alternative. If the current model fails, what does prepare us?
And this is where Dr. Mai comes back in.
Exactly. Because the sources argue the real problem in a major pandemic isn't just the virus, it's the chaos that follows when the system breaks down. Hospitals get overwhelmed, maybe they close doors, people can't get access.
And most deaths happen outside the official count.
Right. People die at home, untested, untreated, uncounted. So the logic is you need an infrastructure in place before that happens, something to manage the load, ensure early isolation, keep things from spiraling completely out of control.
And Dr. Maya is designed to be that infrastructure.
That's the idea. Dr. Maya GPT medical advice you access, it's an AI healthcare aid from Dr. Kadiali Srivatsa, designed to fill gaps, reduce inequality by being accessible, and empower individuals.
How does it work? Symptom checker.
It's more sophisticated than that. It gives free, instant, personalized consultation based on your symptoms using smart devices, but it's rounded in clinical knowledge, scientific standards.
Okay. And how does that help in a pandemic specifically?
The key is its triage system. It's revolutionary because it doesn't just prioritize who's sickest right now. It prioritizes stopping the spread.
How does it do that?
It introduces a new top tier, BLUE priority.
Blue, not red for emergency.
Red is still there for immediate, life-threatening emergencies needing hospital escalation, but BLUE focuses specifically on infectious disease control.
So if your symptoms suggest something highly contagious,
Maya flags it as BLUE. The priority becomes immediate guidance on isolation, even if you don't feel that sick yet. It's about breaking transmission chains early.
that's pandemic thinking built into everyday triage, as the source said. Get people isolated before they spread it widely.
Exactly. Beat the R naught. Then you have the traditional RED for hospital-level emergencies based on combinations of severe symptoms, and below that, yellow and green.
What are yellow and green?
They encourage decentralized care, self-care advice, monitor at home, maybe consult a local pharmacist or chemist. The goal is to keep the vast majority of people out of the overwhelmed hospitals.
So it manages the load, prevents the system collapse, the ghost towns the sources warned about. It pushes decision-making and basic care guidance out to the edges, where people are.
Right. Minimizing those uncounted deaths at home because people have somewhere to turn for guidance, even if the hospital doors are closed.
Okay, but relying on self-reported symptoms, isn't there a risk people could, game the system, exaggerate to get a BLUE alert for time off work, or misinterpret their anxiety as RED symptoms?
That's a fair question, and the answer lies in the AI's design philosophy. It's not just a rigid checklist.
It aims for clinical reasoning.
Yes. It contrasts with that fill-in-the-blank questionnaire approach you sometimes see, maybe with very junior doctors or in high-volume clinics.
What's wrong with questionnaires? They seem efficient.
They can be, but they risk missing the real issue. A doctor focused on ticking boxes might not pick up on the patient's actual most urgent problem, or they might miss subtle cues. It can also feel impersonal, reduce patient confidence.
Okay, I see. Less eye contact, more form filling.
Right. Maya aims for a more dynamic interaction, guiding the user, asking clarifying questions, applying clinical logic to the reported symptoms to arrive at the most appropriate triage level. It's about building trust and getting more accurate input through the AI's reasoning process.
Interesting. So it's trying to replicate, in a way, a good clinical consultation's reasoning process, not just a data entry form.
That seems to be the goal. Which brings us to the absolute core question here. Why now? Why the push for immediate implementation? Why not wait until we identify the next disease X?
Because the whole point is that it needs to be the trusted, functioning system when everything else breaks.
When the traditional services collapse under the strain.
Exactly. The AMR crisis practically guarantees the next pandemic's secondary infections will be devastating. Hospitals will be overwhelmed. Doctors will be unreachable for many. In that scenario, people need an established, reliable system they already know and trust.
You can't just roll out a complex AI triage system and expect public trust overnight during peak panic.
Absolutely not. You need to build familiarity, refine the algorithms based on real-world use, work out the kinks, all before the crisis hits. The infrastructure has to be ready and waiting.
And implementing it now allows for integration with other efforts, right? Like R&D.
Definitely. We saw AI's potential during COVID screening compounds, analyzing data. Imagine having a system like Maya operational globally now. It could feed anonymized real-time symptom data into R&D efforts, helping spot outbreaks faster, maybe even accelerating countermeasure development.
But that relies heavily on trust, doesn't it? People need to be willing to use it, share data, even anonymized.
Critically important. Successful public health AI needs ethical data handling and the willing cooperation of the public. That's why Maya's model, free, instant, accessible, is designed to build that trust foundation now, preemptively. People need to see it as a tool that empowers them, not just surveils them.
Okay. So AI is a key part of the future defense. But we shouldn't forget the basics either.
Not at all. The sources strongly emphasize that even with advanced AI, traditional, non-tech strategies are still vital. Foundational, really. Like
Basic sanitation.
water security, absolutely. Preventing contamination of drinking water seems obvious, but it's fundamental to societal health.
And basic hygiene.
Traditional infection control. Good old-fashioned hand washing, which the CDC still says is often better than hand sanitizers for certain germs. Re-emphasizing simple, customary practices like isolation or quarantine when necessary. Even thinking about materials, like copper surfaces.
Yeah, using naturally antimicrobial materials like copper in high-traffic public areas. These are pre-antibiotic era strategies that still hold immense value. And what about just being outside? The sources mention sunlight, fresh air.
There's historical evidence, like during the 1918 flu, suggesting patients did better with exposure to sunlight and fresh air. Modern theory suggests potential links to boosting the innate immune system through things like melatonin or antioxidants from being near green spaces.
So it's a layered defense. High-tech AI like Maya, combined with reinforcing these fundamental traditional public health measures.
Exactly. Resilience comes from multiple angles. Okay, so let's recap the journey we've taken here. It's been pretty sobering.
Yeah. We started with AMR, that quiet crisis, already killing 1.27 million a year. Then we dissected the current system's flaws.
The illusion of mass testing, the reactive R&D cycle, those incredibly fragile supply chains with single points of failure.
Right. And the policy missteps, the hidden AMR reservoirs in hospitals, painting a picture of deep vulnerability.
And against that backdrop, we positioned Dr. Maya not just as a solution, but as a necessary preemptive measure, shifting the focus fundamentally.
From treating sickness reactively to proactively controlling spread through decentralized triage and community empowerment, that BLUE priority is key.
Because the ultimate stakes, as the source material lays out, are just enormous. If AMR continues unchecked, it makes the next pandemic exponentially worse, up to 10 million deaths a year by 2050 from AMR alone.
Combine that baseline with a novel virus. The level of disruption is almost unthinkable, which really hammers home the urgency. Implementation isn't optional, it's mandated by the trajectory we're on.
Which leads us, I think, to a final thought, something for you, the listener, to really chew on.
We've talked a lot about system failures, broken supply chains, slow testing, flawed policies. But the last pandemic also showed us another critical vulnerability: public trust and belief.
Vaccine hesitancy, outright hostility to scientific guidance. These things hampered the response, even with the technologies we did have.
So the infrastructure, like Dr. Maya, can potentially be built. The technology can be developed. But the final provocative question is, what work do you need to do now? How do you prepare your own critical thinking, your own sources of information?
How do you ensure that you are resilient enough, informationally and psychologically, to recognize and accept potentially life-saving guidance when the next crisis inevitably arrives, whether that guidance comes from an official announcement or perhaps from an AI on your phone?